Hi, my name's Ed Liu. I'm from the Jackson Laboratory. And what I'm going to be talking about is reading and writing in genetic complexity using a single model system, which is the mouse. I'd like to start with what I consider are fundamental truths that my talk will be based on. First of all, that the genome sequencing, particularly human genome sequencing, has uncovered thousands of plausible genetic effectors of disease. Many, or if not most of them, are in regulatory regions, causing some issues around how you transfer that into a model system. The causal variants occur in combination, so you're talking about combinatorics in a grand scale, and expand that to the entire genome of different strains and species, and you have a genetic background problem of a multitude of variances affecting the phenotypic outcomes. Most human diseases need to be modeled in a whole organism. It's quite clear that cells are fantastic and are important, but you can't model Alzheimer's disease in a cell culture. It has to be recapitulated in an animal. Modeling genetic complexity is very hard. Every one of you in this audience knows that. And of course, we are very excited about the applications of CRISPR. At the Jackson Labs, we believe the mouse genetics is the other half of human biology. And of course, there's been some criticisms of the mouse system. It's a failure of the mouse systems in the past because I believe there's inappropriate extrapolation of the mouse as the perfect model system for a human disease, and that's not true. One has to know the limitations of any model system. The modeling has been done frequently on the wrong mutation, a genetic knockout instead of the human variants that are there. Modeling is in the wrong genetic background. We've all had that experience of a null phenotype in one mouse strain that is effective in the other. And when one tests these models, it's always on one genetic background and one mutation because of cost issues. And really, if one wants to transplant this concept of precise model systems, we have to have diversity of both the models as well as the mutations. One of the things we do at the Jackson Labs is to generate new model systems and new systems for testing a variety of approaches. And one of them is actually using diversity strains, where we take 8 founder mice and using very controlled mating procedures, develop what, in one case, collaborative crosses, where they're jumbled DNAs but become inbred species that have very specific rearrangements. The second is diversity outcross, where we can almost outbreed these 8 founder mice so that every mouse in a litter is completely different from the other. In this framework, the entire diversity is in about 45 million SNPs, 2 million indels, and the variances are perhaps more than the difference between the Neanderthal and the human. And you can imagine the differences in phenotype that we can do. But the mouse models 2.0 at the JAX is precise, diverse genetic models of any human disease, which follows this paradigm. We identify all mutations in a patient of a disease. We construct these mutations individually or in combinations in mice, but in this case, either in stable genetic background of a pure inbred strain, or in highly variable genetic backgrounds like a DO and CC, to then test the effects of the genetic background, which we can experimentally extract the components after post hoc. In these, then we do extensive phenotyping. We have just completed the construction of a $21 million phenotyping facility in Bar Harbor, and extracting the cells, individual progenitor cells, like from the brain or from the heart. We can actually do drug screening in cells that we come back to the models and back to clinical trials. In a similar manner, in the genetic backgrounds, we can actually optimize the genetic models because of these modifiers that clearly change the phenotype, improve the stable genetic models that we have, and use these also for drug screening. You've seen many models, many situations of modifiers having dramatic impact, but this is one that was done by Greg Cox for spinal muscular atrophy with respiratory distress, which is a mutation in the immunoglobulin-binding protein 2. The mouse model for this gives you a neuromuscular disorder with motor neuron atrophy. As you can see here, the sciatic nerve is atrophied. But in a specific cross using this, we find that it completely reversed the phenotype. At JAX, we have several of these approaches blocked into a Center for Precision Genetics, where we are looking at patient-specific models, tuned for the exact mutation for the disease, the same mutation in genetic backgrounds, an Alzheimer's Disease Precision Model Center, that we take candidate mutations from human sequencing and multiple mutations in combinations and aging those mice for a phenotype. And in Center for Genome Dynamics, the DO mice is used for super high resolution mapping of quantitative relevant traits such as aging. Now, how do you go about doing this in the technologies? First of all, you can create mice with the same genetic mutation in many different backgrounds. And in our laboratories, we've actually moved the technologies in a scaled manner by having electroporation of embryos, so you can bypass the microinjection phase. The second, which I'll show you a few examples, are creating mouse models with many structural or regulatory variants using a system called Caecilio, which is a combination of Cas9 and Pamilio. Pamilio is a RNA binding protein that has sequence specificity of binding that can be tuned because of the combinations of repetitive sequences that are there in the Pamilio proteins. By engineering that, we can produce a trinary system of a Cas9, a guide RNA, and an effector that is linked to the puff motifs that are there that recognize very specific binding sites, binding sequences in the guide RNA. One can then do multiplexing in a single cell of many different effectors, have multimerization to expand, as well as develop complex formation with multiple protein effectors. In this manner, we've been able to create a few mice with multiple effectors that enhance, activate, and repress simultaneously to actually change the epigenetic code, as well as to actually label cells in multiple loci. Just to give you an example, this is OCT4 and SOX2 simultaneously in a cell using two guide RNAs and the various Pamilio recognition sites. And you can see that activation of OCT4 can occur only when there's when the OCT4 guide RNA is there. At the same time, we can actually downregulate with a repressor in the same cells using a SOX2 guide RNA locked onto a repressor. And you can put those two simultaneously together, and it's highly dependent on having the Pamilio component. So with these kind of tools, we're hoping to develop highly complex models of mouse models of the human condition, even converting many of the loci to human-like features. So thank you very much. Should we have question and answers afterwards, or okay, sure.